For Unit 1, we studied many things, but I would like to focus specifically on evidences for evolution. The one I am showing you in the picture above is homologous structures. First, let me start out by defining what homologous or homology means. Homology means characteristics in related species that can have an underlying similarity even though they have different functions. Now with that, it should help you understand the meaning of homologous structures, which is basically the same structure but different functions, showing evidence of a common ancestor. Moving on to Unit 2, we are going to focus mainly on speciation. The definition of speciation is the process by which new species arrive, or when one species splits into two or more different species. The part of speciation that I have displayed here is the postzygotic barrier called hybrid breakdown. As shown in the picture, hybrid breakdown is when two hybrids mate and their offspring are born sterile or weak. An easy way to remember the definition of hybrid breakdown is calling it second generation breakdown. On Unit 3, I want to focus on the properties of water, more specifically surface tension. Surface tension in water is caused by the cohesive nature of the water molecules. In my picture, I am demonstrating the surface tension of a large body of water. With surface tension, it increases the niche of organisms such as lightweight insects that can stride along, as shown in the picture right here. In addition, surface tension allows small particles to stay afloat instead of sinking to the bottom and destroying our delicate marine life ecosystem. In Unit 4, we studied biomolecules. The biomolecule I will be focusing on today is carbohydrates. A carbohydrate is made up of monomers called monosaccharides. Some examples of monosaccharides are glucose, fructose, and galactose. In addition, carbohydrates contain the elements carbon, hydrogen, and oxygen, and it will always be a 1-to-1 ratio. The functions of carbohydrates are that they are a main source of energy, have energy storage, and are also used for structural support. The polysaccharides that are used for energy storage are starch, such as potatoes, and glycogen, which is especially found in muscle and liver cells. Lastly, for structural support, the polysaccharides that are involved are cellulose, which is a major component of plant cell walls, chitin, and exoskeletons of arthropods, and cell walls of fungi, and lastly, peptidoglycan, which is found in bacteria's cell walls. Now, for Unit 5, I want to talk about passive and active transport. Starting with passive transport is the transport of molecules with the concentration gradient from high to low, which requires no energy. Passive transport includes diffusion, which is gases, facilitated diffusion, which uses transport proteins to help ions and polar molecules travel down the concentration gradient, and passive transport also includes osmosis. Osmosis is diffusion of water, which has three parts or different types of solutions to it. First is the hypotonic solution, or turgid, which means more water and less solute. Secondly is the isotonic or flaccid solution, which means the same concentration of water and solute. Now lastly, a hypertonic or plasmolyzed solution, which is more solute and less water. Moving on to active transport. Active transport is moving molecules against the concentration gradient from low to high. This type of transport requires free energy in order to transport molecules. An important type of active transport is the sodium-potassium pump. The sodium-potassium pump uses an amphipathic peripheral protein, which means it uses an embedded protein with polar and nonpolar ends to actively pump or transport sodium out of the cell and to actively pump potassium into the cell from low to high. Last but not least, for Unit 6, I decided to demonstrate factors influencing enzymes with temperature. All enzymes have an optimal temperature in order to have efficient activity. Although changes in the temperature, whether it's too hot or too cold, can cause the enzyme to denature and lose its 3D shape. As shown in the picture above, when temperature is increased, the heat energy causes more collisions with more energy. So therefore, the higher the temperature, the more collisions, and the higher the chance the enzyme will denature and will not only lose its original shape, but will also lose its effectiveness as well.